Honorable Chairman, esteemed conference participants, before presenting the results of the work conducted, I would like to begin this report with an introduction that summarizes the findings of previous studies and establishes the framework for future research directions. The advancements in nanomedicine open promising prospects for the broad clinical application of officially approved, biocompatible, nanotechnology-based drugs. In Ukraine, the first officially registered biocompatible magnetite nanoparticles for medical use were developed and patented by Andrey Belousov in 1998. These include the intracorporeal nanobiocorrector, ISNB, the magnetically controlled sorbent, MCSB, and the biologically active nanodevice, microMAGE-B. The sorption and magnetic properties of biocompatible magnetite nanoparticles enable them not only to selectively absorb surface proteins of cell membranes through the principle of selective magnetophoresis, but also to prevent oxidative modification of proteins by stabilizing active groups, normalize the state of receptors located on the cell membrane surface, and enhance the activity of membrane-bound enzymes. Previous studies have reliably demonstrated that these nanotechnology-based drugs regulate the enzymatic activity of the antioxidant system in red blood cells and modulate metabolic processes in human leukocytes. As a result of studying the ultrastructure of the lungs, liver, and kidneys, it was found that the introduction of biocompatible magnetite nanoparticles into the cells of these organs induces non-specific activation of metabolic processes, enhances the potential of organelles and the adaptive mechanisms of cells, and accelerates reparative processes at the level of macromolecules and membranes. A previous experiment demonstrated that biocompatible nanoparticles of the intracorporeal nanobiocorrector can be effectively used in MRI studies. These nanodrugs induce reversible changes in the biological fluids of the cellular microenvironment, leading to increased mobility of hydrogen protons, which is the primary cause of altered metabolic processes in malignant cells. This holds promise for the development of new methods for early diagnosis and targeted therapy of malignant tumors. Extracorporeal blood treatment with nanoparticles of the magnetically controlled nanosorbent reliably reduces the activity of calcium-magnesium ATPase in red blood cells, actively inhibits hemolysis, increases ATP and 2,3-diphosphoglycerate activity, restores the bioelectric charge of red blood cell membranes, regulates transmembrane exchange, and inhibits eryptosis. Recent studies have shown that a physiological saline solution, which was pre-treated with magnetically controlled magnetite nanoparticles and subsequently added to preserved red blood cells, actively inhibits hemolysis processes during storage, exhibits membrane stabilizing effects, enhances sedimentation stability, and has a pronounced bacteriostatic effect against pathogenic groups of microorganisms. The optimization of the obtained research results led to the development of a new, simple, and practical method for modernizing preservation solutions, which enhances the functional quality and safety of preserved RBC transfusions. To conduct an in-depth study of membrane structural changes in preserved red blood cells at the molecular bond level, it was decided to use infrared spectroscopy. Briefly about the method. Infrared spectroscopy is a method for identifying and analyzing chemical compounds. The infrared beam is directed onto the sample, and by measuring the radiation absorbed by the sample at different frequencies, it is possible to determine which molecules are present in the sample. The method is based on the absorption of electromagnetic radiation in the infrared range by groups of atoms in the studied object. When exposed to infrared radiation, molecules absorb quanta, whose frequencies correspond to the frequencies of valence, deformation, and librational vibrations of the molecules. Infrared spectroscopy allows for tracking changes in all major types of bonds in the molecules of the studied substances. This slide demonstrates the spectra display graphs showing the frequency and extent of absorption. Since different molecules absorb radiation at specific frequencies and in known quantities, the spectrum can be used for molecular level identification of the sample. I will not go into detail about the materials, methods, and subjects of the study, but will simply present them on the following slides for your reference. It should be noted that magnetite nanoparticles have been produced in the Laboratory of Applied Nanotechnology of Belousov. Results and discussion. Identification of absorption bands in the infrared spectroscopy of the initial red blood cell suspension is presented in next slide. The analysis of changes in the infrared spectroscopy of control and experimental samples in the CPDA1 medium showed that over 49 days of storage. 1. In the red blood cells control suspension, significant destructive changes in the molecular structure begin after 4 weeks, and after 6 weeks of storage, the molecular structure of the red blood cell membrane is completely destroyed. 2. In the experimental sample, after 6 weeks, there is a significant weakening of intra- and intermolecular bonds in the structure of the red blood cell membrane. However, complete structural breakdown is not observed. After 7 weeks of storage, stage EAT, visible disruptions occur in the molecular structure of lipids and proteins in the red blood cell membrane, but some of the most stable bonds are still preserved. Conclusion. The method of infrared spectroscopy made it possible to track the dynamics of changes in all important types of bonds in molecules of erythrocyte membranes at the stages of their storage at positive temperature. The results clearly showed that the presented method of application of nanotechnology significantly increases the storage time of red blood cells in different versions of preservatives due to mechanisms to reduce violations of the molecular structure of proteins and lipids in the erythrocyte membranes. In the future, with used nanotechnologies is planned to continue to study the features of metabolic processes of preserved red blood cells at storage stages at positive temperature. Nevertheless, today it is obvious that the presented method of application of nanotechnology is not only safe for use in practice in the blood service, transfusiology, and hematology, but also is the most promising innovation project. Thank you for your attention.